Welcome to Med in 5 channel. In this video will be discussed about pyrimidine nucleotide biosynthesis. All pyrimidine bases have the same component, the pyrimidine ring. There are 3 types of pyrimidine bases: uracil, cytosine, and thymine. Bases differ each other from the functional groups and their positions. Their nomenclature depends on the structure differences. Here we have the pyrimidine ring. Let's number its angles. 1, 2, 3, 4, 5, 6. Notice the presence of 2 nitrogen molecules in the first and third position of the ring. It is used the abbreviation, nitrogen 1,3. Starting from the pyrimidine ring, we can name each of the pyrimidine bases. For example, here we have nitrogen 1,3, keto, 2,6, pyrimidine. The name of the base is uracil. The second will be nitrogen 1,3, keto 2, amino 6, pyrimidine. The name of the base is cytosine. The third and the last will be nitrogen 1,3, keto 2,6, methyl 5 pyrimidine. The base is named thymine. If the base create compound with ribose sugar and phosphate group, it is provided a pyrimidine nucleotide. Pyrimidine nucleotides are components of genetic material, DNA or RNA. Pyrimidine biosynthesis occurs inside the cytoplasm, and the energy for it is provided by hydrogenated nicotinamide adenine dinucleotide phosphate. Let's explain step by step the nucleotide biosynthesis. Inside the cytoplasm, one molecule of glutamine reacts with carbon dioxide and 1 ADP in order to create the carbamoyl phosphate compound. In the next step, the carbamoyl phosphate will react with the aspartate amino acid. The aspartate carbamoyl transferase enzyme will remove the inorganic phosphate from carbamoyl phosphate and create a new compound. It will be named nitrogen carbamoyl aspartate. The latter is a special compound because it takes a different organization in space. Notice the orientations of the elements. Same structure but different orientation. The gray highlight shows the first position of nitrogen, while the yellow one shows the third position of nitrogen. Afterward, the carbamoyl aspartate will undergo another reaction. The dihydroorotase enzyme will remove a water molecule, 2 hydrogens and 1 oxygen, and will create a bond between the extremes of the compound. Now we have a hexamer structure, named dihydroorotate. In the next step, a dehydrogenase enzyme and NAD coenzyme will create a double bond between the carbons of positions 4 and 5. The substance will be named orotate. In the following reaction, will enter a phosphorobosyl phosphate, which will create the key compound in the pyrimidine biosynthesis, orotate monophosphate. The latter will need to undergo decarboxylation at the 4th carbon position of the ring, with the help of decarboxylase enzyme, providing the uracil monophosphate compound. It contains an uracil base, a ribose, and a phosphate group. Starting from uracil monophosphate, 2 other types of nucleotides can be formed. One option includes glutamine donation of its amino group, and creation of the cytosine 3-phosphate compound. It charges 2 ADP. The highlight shows the amino group. The other option includes methyl tetrahydrofolate donation of its methyl group and creation of thymine 3-phosphate compounds. It charges 2 ADP molecules as well. The highlight shows the methyl group. Now we have all the 3 types of nucleotides produced. That was all from Med in 5 team. Thanks for watching. For other videos like this, please click subscribe.